No, okay. So welcome to Ag Science on Tap for the second installment of our summer semester series on sustainable food production and nutrition. So this is, the second in the summer semester. Last week, we learned about alternative proteins and their challenges and opportunities, or last time, did I say last week? And it feels like last week, and that was from the HSVT. And today we have expertise from the Kompetenzzentrum for Ernährung, or KErn. And Claudia will do the honors of introducing our speaker.
So this is Martin Kussmann. He is head of science at the Kompetenzzentrum for Nutrition. Most people know it as KErn. Please have a seat. And it's a part of the Bavarian State Ministry of Food, Agriculture and Forestry. And for people who don't know KErn is like a platform that brings together agriculture, food production, retail, consumer, science, and education to facilitate integrated solutions for a healthier and more sustainable food system. But he's not only scientist, he's also a CEO and founder of a biotech company. So he combines academic and a corporate career, which I think is very interesting. So we're looking forward maybe to hear something about yourself and about something about your topic, which you can announce again: agriculture and nutrition for healthier people on a healthier planet. So we really, it's an add-on on last time's talk where we already talked about nutrition and alternative proteins. Now we're looking on the whole system. So the floor is yours. Very welcome.
Thank you very much also for coming. More applause when it's over. Huh? So, no, this one. Okay.
Yeah. Okay.
Okay. So anyway, here I am. Returned to Germany 2 years ago, and I want to talk to you about agriculture and nutrition for healthier people and healthier planet. Actually, that's the title I always use, so that I put under that whatever is needed. So I have prepared a little outline for you. I present this with pride as a touristic introduction. This is my little hometown where I grew up, which is near Münster in the northwest Germany. And I know in Bavaria you have tons of castles, but we have that too. So Westphalia is also a very beautiful place. And we call this very proudly the Westphalian Versailles, because this castle has the same footprint as Versailles. It's a little smaller, but we are very proud of this. So I would like to introduce myself, although you already did a nice job, so we can keep this maybe a little short. And then we'll allude to the more general theme of human and planetary life, health and wealth. It sounds a bit like the everything topic, and it is. I would like to address a particular aspect I'm interested in, studying health in its best form, and that's to say human longevity and what it is made of. To dive then more into the hardcore nutrition stuff, about nutrients, bioactives, what we call nowadays precision nutrition or targeted nutrition or systems nutrition, and then wrap it up with a review and outlook. So about myself, you heard basically most of it already. I'm born in Germany, I have Swiss citizenship, and I've spent enough time to New Zealand to still have the permanent residency, of which I'm very proud. And I have various interests besides science, especially nature and outdoors. I'm very much interested in this challenge of today that humanity is facing in the Anthropocene, where we have more influence on the planet than the planet on us, which is a new era with all ups and downs. Not that I'm good at any of this here, but I still try to practice this here. I found a little tennis club here in Freising, which I'm very happy about. And I love to be in other places. I'm notoriously curious, and I had the chance to collect a few languages on the different stops and stopovers during my professional lifetime. I think my ingredients for a healthy work environment are honesty, respect, and sense of humor, and the rest is cultural diversity. This is my credo. And I also believe that there has never been a better time and a more necessary time to do science, and I will come to this later on. In terms of professional background, you already mentioned it. I'm a mixed bag. I've worked in academy, big industry, biotech, also for government to some extent, while I was in New Zealand, professor in Auckland at the Liggins Institute, where we dealt with early life health. This was very meaningful to look into the possibilities how you can get things right in the first place instead of waiting until they go wrong and then try to correct them. So it was all about perinatal nutrition and early life health. I was also tasked with scientifically directing a national science program on food innovation. And this is important, of course, for New Zealand. For those that know this beautiful country, food and agriculture is the biggest economic sector in New Zealand, besides services and, of course, tourism. So this is a mixed experience, which probably also took me then to the KErn, because at the KErn, as you said already, we work with all stakeholders of the food system to make it better. So this sounds, of course, like everyone wants to do that. So what is so special about it? What I like about the KErn is that we work from agriculture to processing, production, retail, like you said, some science, and of course also consumer aspects, to run pilot projects or to exercise communication that demonstrates this is how a food system could look better in terms of sustainability and health, while preserving all the Bavarian specialties, which we all appreciate so much, I guess. Okay. Let's see whether this moves. I mean, it's a beautiful slide, but for the rest of the talk, it's maybe a bit boring. Okay. I would like to start out with a graphic. Maybe it's not, you can't read it so well because it's a little out of focus, but I think should be fine, of this book Factfulness that probably at least the older of us may know quite well. It's a beautiful book about how the world is and how the humans see the world. I put this up because I think we live, of course, in this era of Anthropocene, with all these challenges, but we also live in an era where we have this impression the world gets worse and worse, and everything gets worse and worse. And that's not true. So what I'm showing here is basically all bubbles represent countries. The size of the bubbles correspond to the size of populations. And there are two very simple ways of measuring how well people are doing. And this is how healthy they are, and you measure this simply by longevity as one outcome, and how much they make. So how wealthy they are, how healthy and how wealthy they are. And the take-home message of the last 50 years is that every bubble has moved from bottom left to top right. And the gap between the poor and the rich countries is also closing, which is often reported to be the opposite. So this does not mean we have a lot of work to do, and I will come to this, but the world, in my view, has never been as good as a place as it is today. Now we know we have this issue that we are consuming more than one planet per year. And we just had the Earth Day. You don't have to be a sustainability expert just to realize that if you consume 1.7 planets per year, that's in the long run, it's not gonna go well. So we have this sustainability issue, and I think we have two. We have the sustainability issue of energy and food. These are the biggest challenges probably we have. We must generate energy and food in a more sustainable way. And with food, of course, there comes more to it. And I explain this later. So we need nutrition for healthier people. This means we need more sustainable healthcare, not only repair, a lot more prevention. Disease prevention, especially because the lifespan has increased so much over the last, especially half century or century. Life expectancy is around the 90s these days in developed countries. And we would like, of course, to match this with a concomitantly prolonged health span. This is, of course, a very big challenge to achieve. And we need this of course, on a healthier planet. And this alludes to the fact that we are now using this concept of One Health, which basically means there's no such thing of healthy people on an unhealthy planet or vice versa. It can only be integrated. It can only be looked upon as an integrated solution. And we know that we have soon, now we are about 8 billion, soon we will be 10 billion to be fed, which poses enormous challenges, especially for protein supply. Calories, I think we're still doing okay. But we also need to think about the quality of the nutrition, not only the calories. We have the dichotomy of overnutrition calorically and undernutrition in terms of micronutrients. And we also know that we have a lot of impact on biodiversity, and we promote climate change. This said, the planet can feed 10 billion people. It's not that Earth can't do it. We just have to do it in a different way. And we do not have so much, I think, a production problem. We have a conversion problem, a distribution problem, an estimation problem. So we can produce food for all these people, but we have to do it in a different way. Now I would like to start to say something again positive, because I'm a notorious optimist despite all these challenges, something positive about agriculture, which is incredibly under scrutiny these days. If you listen to the press, it seems like they put poison in the soil and produce the wrong food, and the tractors make everything flat. And I mean, this is just, sorry, this is a scenario which just doesn't apply. Agriculture has, in my view, lived a tremendous success during actually almost my lifetime. I'm born 64, so I just turned 60 2 weeks ago. And across this short period of time, the population grew from 3 to 8 billion, and the amount of people that being undernourished, which is still very regrettable, remained constant. So this is a tremendous success. Of course, now you can say, well, it comes out with all the damage and all this thing. Well, we could also have ecologically let all these people die, right? So to be cynically, to express it cynically. So this is a tremendous success. And by the way, the main reason for now being 8 billion versus 3 billion not so long ago is reduced child mortality, much more than extended lifespan. Lifespan adds to that too, but especially because children could survive. And this is, again, mainly due to the fact that infectious diseases are much better controlled these days, okay? So that's just facts. And at the time, one farmer could nourish about 17 people. Today, one farm nourishes about 140 people. So the productivity is enormous, but it comes at a cost. So genetic diversity has been dramatically reduced in order to optimize yield. Everything was geared towards more yield. And this has, of course, industrialization of agriculture has led to deforestation. We lost and we are still losing biodiversity. We use enormous amount of water for this. We have also contamination issues. The soil is suffering. We have to deal with waste. So there's tons and tons and tons of problems. But don't forget, we can feed 8 billion these days, almost these days. So now the message here is it's a success, but business as usual is no longer applicable. We need to change. And especially the food system must change, because it's not only agriculture that poses problems, we also have other aspects in the food system. And I would say this is the worst here. This is about 1/3 of the calories basically go wasted. So only 2 calories of 3 are eaten. The rest is lost from basically the field to the consumer somewhere, in production, in transportation, and spoilage and whatsoever. So as long as we don't reduce food waste, and much of this is actually happening in private households because we buy too much and throw away stuff, as long as we don't deal with this, I think we can forget about all the rest. I mean, a system that produces so much waste and is so inefficient, this is primordial to be addressed. And of course, you know that everybody thinks about this, how can we do this? And the Eat Lancet Commission, I don't have to go into details because you're probably more familiar with this than I am. They have come up not only with an analysis of how the planet could feed 8 to 10 billion, they also have come up with a suggestive diet that would allow sustainable food production without people suffering from having too little, right? So this balance was tried, they have tried to balance this out. Produce food in a sustainable fashion, don't overuse resources, but at the same time, people should have enough also, not only calorically, but also from a nutrition standpoint. So this was a model, a model that has been established. And now the art lies in the way, how do you translate this global recipe to the different regions of the world? So you can't just copy paste this, right? And this is, in all modesty, our role also at the KErn, to think about how can we take this input and make the Bavarian food system better without erasing everything and replacing it by something completely different, with respecting the culture, respecting the diversity that we have, and all the special conditions that we have in Bavaria. And I think this applies for every region that we need to deal with this. Now, I had the pleasure and privilege to live 15 years in this place, not in the vineyard there, but near Lausanne, Lake Geneva. So if I have a home somewhere, I would say it's there, also because my sons are born there. And I still miss it to some extent because it's so terribly beautiful. And it has basically everything it takes. But I titled this slide Relevance of Research and Solutions. And I explain you why. My next slide is this. And I just want to say that we Europeans, and especially us Germans, we are very good in telling the rest of the world what they should do and what is good for the planet, for the people. Very good at that. I don't know whether this is residual colonial arrogance or so in Europe, maybe, but we are very good in preaching and teaching the rest of the world what they should do. It matters where you live, okay? So you can, of course, promote going back to ecological farming and have a biodiverse garden and all these things. When you live in an area like this, fine. This is beautiful, right? But this is not a relevant situation for the world, because this is where people live. About 3/4 of the world's population live in these big cities with 5, 10 million or more. So now you can say, oh, well, this is so terrible, and well, this is a fact, and I think it will not change, because this is where the money is and where the wealth is and where the progress is. So people will, to a certain extent, still move into cities, and we will have these cities. So we also need to think about solutions that apply to this. And I heard that you have a program here on urban, what is it? Urban productivity, so kind of urban farming and that kind of thing. So you need to think context, right? And not only think in these idyllic terms. So let me spend a few words on human longevity. We know that the phenotype, now we really switch gears, the phenotype of any of us is the result of the genotype and the impact of the environment. This means the genotype is basically your potential, we could say, and the environment are the conditions of what you can make of this potential, okay? And that results in the phenotype. Now, human longevity is a beautiful thing to study because it's a good positive model. Everybody studies disease. The National Institute of Health in the US, for example, is actually de facto the National Institute of Disease. It's not about health. No health institute is about health. I mean, most of these things deal with diseases, so with things that go wrong. So you also should study health when it's achieved, especially at older age. It's a beautiful model to understand how could it go right. And if you look at the 5 blue zones, and there are probably more on the planet where people become particularly old and they stay healthy, this is a selection. So there's quite something is about the Mediterranean area. So there is in Greece, there is an island where people get so old, and of course they have this Mediterranean diet, which has been praised for decades for its health impact. But then at the same time, Okinawa, that you probably know, a southern Japanese island, is also known for this phenotype, totally different environment. People have a healthy diet in the sense that they eat a lot of legumes and here potatoes and other things. Here we have, again, a region in Sardinia, people who have been to Sardinia know that these people do not necessarily consume a diet that you would intuitively consider as healthy, because they are carnivores. On Sardinia, there are a lot of carnivores also, because Sardinia is a continent in itself, and historically, for the Sardinians, the ocean was hostile because it was all the enemies that came, right? So they stayed within the island. So they're not seafarers, it's the opposite. They get very old. In California is such an island, and last but not least, in Costa Rica, which is a completely different environment. Now, Costa Rica, you can say, well, that is easy. I would travel to Costa Rica next month, and my girlfriend stems from there, so she will show me how it is like. So yeah, you can say, well, it's so, you must become 100 years old because the food just grows into your mouth while you sit on the balcony because it's so fertile and everything is so healthy. Anyway, the typical scientific question of all this would be, oh, that's great. Now we know how longevity works. We just have to find out the commonalities, and then we apply it everywhere, okay? It's the completely wrong question, in my opinion. And this is why I showed you in the beginning the slide, phenotype is a result of environment and genotype. When you consider these 5 blue zones, everything is different. There are no commonalities. There are some commonalities. People move, and they don't eat extremely unhealthy food, but all the rest is different. Climate, the soil, the food, geography, and especially genetics is completely different. So we need to think about this interplay. So there are many ways of, becoming healthy, long-lived, and healthy. It's about these gene-environment interactions. So now let's now talk food, and especially about nutrients and bioactives. And now it becomes a bit more molecular. After all, I'm a biochemist, and I like still molecules and to measure things. We have come up with a special issue in Frontiers about the challenges of humanity in the Anthropocene and what it poses in terms of the food system. And one aspect we highlighted here was I think we should much better exploit what nature has to offer in terms of bioactives. So nature, I think, has a solution, a molecular solution for almost everything we need. It's just so difficult to find them because the complexity is very large. This is actually one of the reasons why Merck at the time went to Costa Rica. They bought a square kilometer or 2 of the rainforest, and they grinded it all up and tried to find bioactives there. It wasn't very successful. It was a kind of a brute force bottom-up approach to find good molecules for good purposes. Anyway, this is now probably difficult to read, but what bioactives basically means is classical nutrients, macronutrients, micronutrients, but also these so-called phytonutrients. So not classical nutrients, but components of plants that are health beneficial. And then we also have those things that regulate our microbiome, which we now know is integral part of our health, simply because it's such a big ecosystem. And honestly, the microbiome, I think, is nowadays praised and blamed for pretty much everything we are and do. I think it's a little bit exaggerated, but it has certainly a strong impact. So this, I think I can skip, right? Because that saves you 10 minutes and you can have another beer and you ask your questions. And so I will skip all this because you had a full talk about this very recently. Just to say, protein is an issue, and we need to see how to bring about protein from different, more sustainable sources. We cannot feed 10 billion by continuing consuming steaks and burgers and sausages as we do. That's very clear. So we need to think about that, and there are many, many ways of possibilities, and you can look up these references. So that saved me quite some time, which is good. And now, before wrapping up, I'd like to come to this term precision nutrition, and you can, as I said, replace it by personalized nutrition, targeted nutrition, systems nutrition. Now, if you ask all these specialized scientists, they will say, oh, this is very, very different. It's not personalized, it's precision. For me, it's all semantics. But basically what it means is you take the biological system of the human body and the molecular system of food in a holistic way, and you look at it as a system. So you do not necessarily do the reductionist approach, which has been traditionally the case in the older science, so to say, where you basically dissect the system to its individual pieces, and you study a bilateral relationship, keep everything constant, and then you say, okay, if this goes up, this goes down. But then by doing this reductionist approach, you do not necessarily understand how the system works. It's like if you disassemble a car and study each and every piece, you know the pieces of the car, and you may know what this piece can do, but you still don't understand how a car works, right? So this is all about precision nutrition or systems nutrition. This has, of course, to happen in a cultural context and in a societal setting. So we have here first the individual, and we are all very, very different in terms, of course, our biology here about the socio-demographics. We have the social environment, the physical environment, and also the macro environment is basically the society or the country in which we live. And all these factors impact the food system. And you can apply this exactly the same, for example, to Bavaria, right? So we believe that we need to study or to integrate or to consider at least all these different elements to have finally an impact, because in the end, it's us that decide what we eat, right? And what we purchase. And we can preach and teach until the cows come home. If there is no attractive solution, you would not eat anything, not drink anything if it tastes bad. That's a killer. Or a yogurt could be as fantastic for your health as ever. If it costs $5, no, it doesn't work. So this has to be looked into in an integrated fashion. And now a bit more into also the research that I've been involved in. And I would just like to address a few things here about nutrition clinical trials. And this probably accounts for many other clinical trials too. What we basically do is we look at differences in averages, right? So everybody knows these box plots. So you basically take a group of people, phenotype them, and you say, you're the group of the cases and you're the group of the controls. So you're disease A or you're disease B, or you're old or you're young or whatever. And then you apply an intervention or you do an observational study. And then you look at the group results, you average everything and say, oh, great. The two clouds are slightly different, right? So there's a difference. The issue is the distribution within these clouds is also very wide. And there may be individuals that are negative responders and positive responders in the same group. So this is basically schematically the response distribution across individuals. So what we basically need to do is we need to much more look at individual responses and individual trajectories, just to basically take you as your own case and control. That doesn't mean you need to also look at the group, but you need to monitor what is your trajectory. Do you go up or do you go down? Because after all, none of us is a mean or median. We are all different. So that is a principle that we need, I think, much more to apply. And we call this aggregation of, so to say, N of 1 studies, where you have individual trajectories, and then you try to pool those trajectories that are similar, and then you can draw conclusions for larger groups. Same it is with genetics. We all like this case where there are rare alleles that are causing Mendelian disease with big effects. So rare genetic variances causing big effects. Unfortunately, this is the reality, especially in nutrition. So we are dealing with common variants, very frequent across the population for most phenotypes, which have individually very little effect. So this old idea, as you may remember from the Human Genome Project, where we say, oh, great, we sequence the genome, and then we know what is a sick gene and what is a healthy gene. It just doesn't apply. It's all gene-gene and gene-environment interactions. That makes the whole story so complicated. And then nutrition has even an impact on epigenetic phenomena, just to briefly allude to the 1-carbon pathways in the body. And we know that some food sources are very prominent donors of 1-carbon building blocks. So what you eat can also influence your epigenetic profile. And epigenetics, just for everybody, if I'm too trivial, you tell me. But epigenetics is basically, the word says, is above genetics, so it doesn't concern the sequence. It's basically how the genomic information is packed up. And epigenetics controls which genes are read in which context. So it's the packaging, basically. And it is influenced by nutrition. A few challenges and solutions to bring this precision nutrition concept abroad. As I said, clinical studies are still a big challenge because, first of all, if you go to the literature, many studies are not comparable because rather than applying the same or comparable translational study designs to cohorts of different ethnicities, we did the opposite. We did various study designs to the same ethnicity, like us, or at least me. I know we are already a little bit of a blend of ethnicities, but let's say Caucasians, because this is where the money was, and we just couldn't be bothered to test other ethnicities. So it doesn't make sense to, if you want to find out what is good for which population or which population group, you need to have comparable designs and then see what happens in cohort A, what happens in cohort B. Does it work for those or not? I've been living in New Zealand, for example. It's a perfect example. I haven't met a single slender, no, two over the years, two slender Maoris. All Maoris were wide as this table. Terrible. Because, well, I mean, these were Pacific Islanders that have been living on plants and fish, basically. And then they were, of course, all of a sudden exposed to Subway. That doesn't work. And then also there's the money issue. In New Zealand, 6 apples are more expensive than 3 deep pan pizzas. So there you have an issue. And they were so over-concerned and affected by diabetes and obesity. It was striking, right? So we need to consider this. The meta-analysis are often not possible because the reporting of the studies is just qualitatively not sufficient. I already addressed this. Nobody is a mean or a median. People are superficial, or subjects are superficially phenotyped and then binned. We should not bin you in a group. We should first measure you, run an intervention, and then decide to which group you belong, and not the other way around. Data is a big thing. Everybody, of course, wants data, but data doesn't equal information. Information doesn't equal knowledge, and knowledge doesn't equal wisdom. So we have a lot of data cemeteries, I would say, where we store a lot of stuff, but we can't retrieve it. And then we also know that the data that are not so positive maybe not be published because you can only, promote your career with positive data. No one gives you a research grant when you write a paper entitled, "This actually did not work." Very difficult to find money for this. So that is an issue. And then, of course, the biomarkers, this relates back to the phenotyping aspect that I mentioned. Clinicians and especially regulatory authorities, they like single or very few biomarkers. They like CRPs for inflammation, and glucose goes up or something simple. We are not yet used to, let's say, profiles of markers that give you kind of a prospective health indication. But this is actually where we have to go. And I would argue that in the future, when we have our medical checkup, it should be more possible that some of these things are measured individually for you, right? Not just a standard panel, but it should apply for you and maybe for a larger part of the population, but it's more specific. And then, of course, technology. I mean, I'm an omics guy, as you probably have noticed. I used a lot of these machines, these mass specs, mass spectrometers, and NMR spectrometers are much more expensive and complicated than sequencers. But whatever it is, most of this technology is still residing in research environments. So what we then need is we need to use this for discovery, and then establish a marker panel that has value for a certain consumer or patient or population group, and then translate it into an assay. And we all know how difficult this is because it has to be also economically compatible. So in the end, medical doctors should be enabled to use a device because they don't have mass spec, right? So you have maybe a little ELISA or something. And then eventually you should even have it on your, don't have it here, on your iPhone, right? I mean, now sensors are coming along and you could, then measure yourself. This is I think the long-term perspective. We also use this is a work we did in Auckland. We use now you can talk about this, you use the omics and do all these biomarker studies, that's all fine, but can you do this at large scale? And does it mean anything? And we have tried to promote metabolomics to what we call establish these so-called precision phenotypes that have a more refined profile of a, let's say, a molecular readout of people that is more indicative as a simple test. And as an example, we looked at population level, how plasma B vitamins that are very important for health behave in parent-child diets in pairs of parents and their children. And we were asking the question, how much does the common environment matter? Because typically children live with their parents. Genetically, there is some relationship, but then we also had different environments. So we were trying to dissect what does influence these vitamin levels. And then you get, for example, just to give you some ideas that I'm not only talking and showing text, but we also produced quite tons of data. We did this on, I think, 1,300 pairs. So that's a pretty big deal to measure all this. And we did it across multiple vitamins and micronutrients. And then you see here the distribution curves where the light or the turquoise color, that's the parents, and these are the children, the dark blue ones. And here is concentration. So you see distributions, and then you can dissect what is more important. Is it the genetic ancestry? Is it the common environment, and so on. We also did some interventions in what we call community-based participatory research, very bulky term. That is an example of a nutritional intervention that does not take place in clinics, in a sterile environment. It takes place in the field, so to say. So with my friend Jim Caput and Jacqueline Montero, they went to Brazil, and Jacqueline is actually a professor in Brazil, and they looked at children in 2 different years, so about 150, 9 to 13 years old. And then they were assessed at baseline. We did the demographics, puberty stage, and genomics. And then there are different visits where we threw all these measurement technologies we had, metabolomics, proteomics, whatever we can get hold of. And we looked at the kids before and after an intervention. And the intervention was a multi-nutrient intervention. It was a little tablet, which contains, well, I think it was like 6 or 8 micronutrients that are important for health. And we asked the question, does this supplementation make a difference to the health of these kids before and after? And we replicated the study, which usually in science rarely happens. If you have a good result, publish it and don't replicate it, because it's very difficult, especially when it's about health in humans. So we did that, and then we did not only the population-based analysis like here, but remember that schematic slide I showed before with the non-responders, the basically neutrals, and the positive responders. So we looked at each and every individual in order to understand how does this intervention work and what kind of effect does it have. And this becomes even more complicated, especially at this resolution, but don't worry about it. I just want to, I will take you through this. So this top panel here shows you the genetic admixture of the Brazilian population. Okay, so there is an average background, and then you see spikes here of in different colors of African, Native American, Brazilian. This is, I think, Asian here. This is LATAM, European, whatever. So we did basically a genetic analysis of all these subjects in order to say what kind of ethnicity are there predominantly, or what kind of admixture do they have. And so Brazil is one of the worst case scenarios, because usually nobody, I mean, you know Kari Stefansson, right, in Iceland. He did this early genomics project because Iceland is such a nice example. 250,000 Vikings, and they're all the same. Genetically, everything is so similar. So this is the opposite of Iceland. It's lots of people, and people are from all over the place. But we explicitly did it in order to understand, can we apply the systems nutrition approach to understand, does genetics actually matter for 2 things? Baseline nutrient levels and response. And what you see here is that, of course, the dots are quite dispersed still, but these are baseline vitamin levels. Now I can't even read it myself here, depending on the impact of the different ancestries. So this is, I think, maybe European. I can't read it here. Anyway, the principle is you look at baseline nutrient levels and response of these nutrient levels depending on genetics, okay? And this is going into personalized nutrition when it comes to nutritional intervention. 10 minutes, I'm done. Is it okay? Or are you exhausted? I am exhausted, but I need a beer. But usually, as I said, once I get going, there's no hold. So, but I'm happy to also, of course, answer the questions. Everybody has AI on the slide nowadays, right? You can't get funding if you don't put artificial intelligence, of course. And everybody pretends to do it. I had the pleasure to work as a CSO for Nuritas in Ireland. There's a friend from Ireland. We had already a little chat. Beautiful place and wonderful people. And I worked with them because this was a company that tried to predict peptide function purely by computation. And so how does that go? So in principle, the idea was to predict as much as possible in order to avoid all the unnecessary testing, okay? Because usually when you test bioactives, you do this brute force screening, you do lots of assays and fractionation, and 99% doesn't work and you throw it away. So it's a brute force approach. So the idea is, can you actually know beforehand what is a good candidate? And so what we did is we asked this question. We wanted a health benefit to be achieved, for example, muscular health. We wanted to avoid muscle atrophy as an example. And we had a huge knowledge base of peptides, what they do and how they look and what kind of properties they have, okay? Now I'm going to explain you how this works. So this was basically you take in all public knowledge that is available. And of course, you produce proprietary knowledge because you do all this testing in-house. So you basically build a brain that knows not much, but everything about peptides. That's for sure. You can ask this brain about peptides. So how does that work then? And how do you get answers? So here you can see clouds of peptides that have been projected in a three-dimensional space here according to principal components 1, 2, and 3. And you know, the red dots, these are peptides that are good for blood pressure control. They're anti-inflammatory peptides, antimicrobial. Some are good for glucose regulation, anti-aging, and so on. So I use this illustration to explain you how this works. So you have to imagine you have a library of knowledge for peptides, okay? But how do you find the book and the page where your answer is written? So you can imagine that every peptide has a three-dimensional structure, right? And you can put, according to the three-dimensional structure, you can put the peptide into a three-dimensional space with all structures, and every peptide would occupy a single spot because of the structure is unique, okay? So the only thing you have to imagine is instead of only having 3 spatial dimensions, you have 100 dimensions that cover all peptide properties. So this cube is a multidimensional space, and each and every peptide occupies a particular spot in that space. And what you then do is you drill a hole into that cube from the angle and the depth that represents the properties that you would like to see in that peptide. This is how you can imagine this, you know, knowledge base. So this is what we did and what they did. And you can interrogate this multidimensional space, and then you can predict and test and validate these peptides. And the easy thing is actually to blast them against the plant and food proteomes, because that's a simple genomic analysis. That's the beauty of using peptides for nutrition, because these are the only nutrients that are directly encoded into the genome, right? All the other nutrients are secondary products, and it's much more difficult to predict them directly. And we tested this, so we predicted a blend of peptides that should avoid muscle atrophy and reduce the muscle inflammation that you would have, for example, when you have an injury, you don't move, or also when you get to older age, you know, and everybody, I mean, as I said, I turned 60, so I'm a good client for this, because from the age of 30 or so, your muscle performance goes down and you're losing muscle mass, especially if you don't exercise. So I was happy that I was not part of that trial, because it was pretty heavy here. So we recruited men and casted them one leg for a week. And it was also the walking in circles trial called, right? So this is what we call it, because they could only walk like this. And then the beauty of this trial is, again, think about case and control. Nobody is immune. This guy is its own case and control, because we could take samples from that leg and from this leg. Okay, that's cool. And then you can then ask several questions. How does the leg recover? This was casted. How does it behave compared to the other leg? And you can use, for example, like what you do in fitness centers, you do a leg press exercise, you can measure inflammatory markers. And to make a long story short, our mix here, which we call NPN, this stands for Natural Peptide Network here in blue, and we compared this to a milk protein concentrate that is sold, for example, in fitness centers. It's basically easily digestible protein with no specifically designed peptides. It's just a nice blend of easily digestible protein. We compared this and we could see here, although the baseline was of course different, but that's not the point, that here with this supplementation, the subject could regain the full leg press strength as before the trial, whereas this group only could partially recover. So this is just one of those examples, okay? So my point here is you can do a lot in silico before doing the experiments. And remember the previous slides when I talked about bioactives, if we use smartly this artificial intelligence and ask these questions, what is it good for? We can use that for basically more efficiently mining the biochemical space that is out there, which is still largely underexploited. I'm coming to an end. I would like to thank a few guys without who I would not be standing here, and I pardon for the gender balance, it's all guys, but that has just happened. This is Peter Röpsdorf. He's a Danish Viking. He was my postdoc supervisor, one of the most unusual supervisors you can ever meet. He was rolling, I don't know, his 20th cigarette when he was recruiting me and said, Martin, the way you behave, you can well join our group. That was his interview. I was just trying to get a grant to go somewhere else. And I said, oh, if this big shot is so nice, maybe I should go there. And apart from the weather, it was a wonderful place to be in Denmark. Then I met Danny Hochstrasser, who is the father of clinical proteomics. I worked with him in a proteomics startup at the time. We were the biggest in Europe, at least. And then we found out how difficult human proteomics really is. If we had known before, we would maybe not have done it. But it is what it is. And Laurent Fay was the head of bioanalytical sciences at Nestlé, and he brought me to Nestlé. And on from then, in the early 2000s, I moved into nutrition because I found this idea so good to use nutrition in a much more efficient way to promote health. This is probably my best buddy in science. We still work together. He has a personalized health company in the US. Meanwhile, we were together at Nestlé for quite some years. Jim is, I would say, the father of systems nutrition. He coined that term, the holistic view of nutrition and human health. Jose is a professor at Tufts in the US and also at the India Institute in Madrid. And we had a wonderful time together and met at conferences and things. And also Pierre Deschlot, I would like to mention, he is professor and MD at the Université de Rouen in northern France. And all these people I owe a lot because they influenced my way of life, I would even say. And they were there in good and in bad times, which is probably what you call a good friend. I think I promise you to come back to this introductory remarks. There's never been a better time for science. And this is, of course, addressed to the younger people here that still have all their life ahead of them. So I can only say my boys are in science too, so I don't know how that happened. But if I didn't have to make money, I would do it again. I would subscribe to Life Science Today. Jonas and Adrian, they have both been in Berlin. One was first at the EPF Lausanne, and the other one is now at ETH Zurich. It's so much fun, and you can move, at least within Europe, very easily. And so it's fantastic. And if I have just a very generic advice, I mean, I would really follow your intuition. And I think being mobile is a key. And then for me, it was always important to also find a place where I felt, oh, this is a pioneering place where, you know, they are maybe a bit ahead of the curve, and also the relevance. So I could also have, for example, studied protein structure and do protein amino acid mutation and still look whether it still folds honestly, I couldn't care less. So I always was interested what comes after the paper, because papers are the academic currency, but you can't eat them and they don't heal anything. So in the end, it's all about how you translate it. I think interfaces are important. I never cared too much what it says on the door sign, whether it's institute or now it's the KErn, it's even part of the ministry. I would not have thought I would ever be there. Or it's big industry or biotech, it doesn't matter. There are always places where you will find, oh, this is a hotspot, and this is good to be. And of course, it's all about building and maintaining good relationships. Also, when you don't need something, you know, many people are good in networking when they need something, and when they have it, they disappear. You notice. So it's good to it's good to entertain relationships over a long period of time. And then, of course, if you can choose or have the luck, like me, to have good mentors, that this helps. Science is not an alternative fact. That's the coolest license plate I ever seen. This by my friend Jim Caput. And I just put this up because this is my last slide. I think we all have a big responsibility these days. There's so much bullshit, now I say it, around. You know, you find yourself in discussions where people say maybe the Earth is a disk, right? And maybe it's not a sphere. Then it becomes very difficult to have a rational discussion. And we all know what social media, of which I'm a fan, it's just about intelligent use. It's all good, but it's so easy to put crap out, and it's so difficult to compete with quality. And I think, especially the younger generation, I can only encourage you to do what you do. It's so relevant, it's so much fun, but you also have a responsibility, and I can only ask you, speak up when you hear bullshit. Thank you very much for your patience, and I'm happy to take questions.